In the coming weeks, we're going to be discussing three new sets of biochemical reactions. We are going to talk about transcription. We're going to talk about translation. And we are going to talk about DNA replication. Now, transcription is the synthesis of RNA, which is a polymer. It's a nucleic acid. It's a polymer. Translation is the production of protein. It's a polymer. DNA replication is the production of, as you can guess, DNA. It's a polymer. So all three of these are polymers. RNA and DNA are nucleic acid, and proteins are proteins. And what that means as a polymer is it's simply taking monomers and covalently linking them to one another. Now, the difference here, there's a fundamental difference between these polymers, nucleic acids and proteins, and polymers that we've looked at before. And this fundamental difference really changes some of the aspects, some of the things that we need to think about in the process of transcription and translation and DNA replication. So let me see if I can make that clear. So let's take a look at glycogen. Right? Glycogen is a polymer we've already looked at. Glycogen is a carbohydrate, a polysaccharide, and it is made up of nothing more than glucose connected to glucose to glucose. And just like what we're going to see for those three processes that I just illustrated, DNA replication, transcription, and translation, we have an enzyme here that's going to catalyze it. And the enzyme here is called glycogen synthase. So glycogen synthase. And this glycogen synthase is adding glucose, and glucose, and glucose. So as it's building this polymer, after it's done adding one of the monomers, one monomer of glucose, how does it know what the next monomer is going to be? Well, the answer is because there's only one thing that it needs to know. It's glucose and glucose and glucose and glucose. That glycogen synthase doesn't need any external source of information. It is built to do nothing more than add glucose to glucose to glucose. The only molecule that will fit into its active site is glucose. So what that means is this molecule here, this polysaccharide, that's only made up of one repeating unit, has no information in it. It's just glucose, 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 and therefore glycogen synthase, the enzyme, doesn't need to have any external source of information to figure out what the next monomer is going to be because it's always going to be glucose. Now let's take a look at the production of RNA. So that is transcription. And it's the production of RNA, okay, which is a nucleic acid. And nucleic acids, specifically RNA, are made up of various subunits like A's and U's and G's and C's. And the enzyme that catalyzes that is RNA polymerase. So let's say we have the beginning of an RNA molecule. We're at 5 prime here. Remember, you call the ends of nucleic acids the 5 prime end and the 3 prime end. And maybe we have A, U, C, and now that RNA polymerase wants to add another one. What is it going to be? Is it going to be a C? Is it going to be an A? Right? How does that RNA polymerase know what the next nucleotide is going to be? Because every RNA molecule is unique. It has to be because it encodes very specific proteins. And the order of the sequence of the nucleotides in RNA is going to dictate the sequence of amino acids and proteins. And the sequence of amino acid and protein is going to dictate the structure of that protein and therefore the function of the protein. So the sequence here is super, super important. And the question is, how does RNA know what the next nucleotide is going to be? And the answer is template. There is a template strand specifically for transcription for RNA polymerase in the DNA that RNA polymerase is going to read. Oh, that should have an E there. That's where it's going to get its information. But that DNA is huge, right? DNA is really long. And that RNA polymerase isn't going to simply read all of the DNA. It's going to read a gene. But the question is, how does the enzyme know where to start reading? Because it's not going to read here. 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 It's going to start right here. And then it's not going to just keep reading to here, to here, to here. It's going to end here. And so when these enzymes that catalyze the production of polymers with information, that have unique sequences, that require an external source of information in the form of the template, they have to have the template, then they have to know where they're going to start the reaction, and they have to know where they're going to end the reaction. They have to know where that reading is going to start and where that reading is going to end. So, as we go through transcription, as we go through translation, as we go through DNA replication, that's what you have to ask yourself. You always have to ask yourself, what's the template? What is that enzyme reading? Then you have to ask yourself, what determines where on the template it's going to start reading? What determines where on the template it has to end reading? Because it's not going to read the entire template. It only reads at a very defined start and a very defined end. So, with each one of those processes, keep those questions in mind. What's the template? Where does it start reading that template? Where does it stop reading that template?